Welcome to G Fun Facts Online. Today we are turning our attention to the arid landscapes of Zimbabwe and Mozambique to examine a biological phenomenon that is reshaping our understanding of time, mortality, and the very nature of aging. We are talking about the African turquoise killifish, scientifically known as Nothobranchius furzeri. To the casual observer, this small, vibrant fish might seem like just another aquatic inhabitant, but to the modern biogerontologist, this creature represents a vertebrate time machine, a miracle of evolution that compresses the biological decay of a human lifetime into a matter of months. To understand the significance of this fish, we must first look at the environment that forged it. The killifish does not inhabit the stable, permanent waters of great lakes or flowing rivers. Instead, it calls the ephemeral ponds of Southeast Africa home. These are seasonal pools, formed exclusively by monsoon rains. They are transient bodies of water, existing for perhaps 4 to 6 months before they evaporate entirely into the cracked, baking earth of the dry season. This boom and bust ecological cycle places an immense, crushing evolutionary pressure on the killifish. It is locked in a frantic race against the sun. From the moment it hatches, it is swimming against the current of biological time, destined to grow, reproduce, and die before its home vanishes. This environmental urgency has resulted in a life history strategy that is unparalleled in the vertebrate world. When the rains arrive and fill the dusty hollows, the killifish hatching is explosive. The fry burst from their eggs and begin a growth spurt that defies logic. In a mere 2 weeks, these fish reach sexual maturity. To put that in perspective, a feat that takes humans over a decade and mice several months, the killifish accomplishes in 14 days. They mate voraciously, laying drought-resistant eggs into the mud to secure the next generation. But this strategy, known as live fast, die young, exacts a steep physiological price. That price is rapid aging. By the time a killifish is just 3 months old, it is effectively a geriatric patient. The decline is not subtle, it is systemic and catastrophic. If you observe a 3-month-old killifish, its once bright colors have faded. Its spine begins to curve, a condition known as kyphosis. It loses muscle mass, a process called sarcopenia, mirroring the frailty seen in elderly humans. Cognitive functions decline, cataracts cloud their eyes, the immune system falters, and they become prone to developing cancer. In essence, the African turquoise killifish experiences almost every hallmark of human aging, but it happens at warp speed. It allows researchers to observe the entire process, from birth to natural death, in the time it takes to complete a single semester of university. For decades, the field of aging research was dominated by 2 primary models: the laboratory mouse and the nematode worm, C. elegans. While these models have been invaluable, they possess significant limitations. Mice are biologically close to humans, but they live for nearly 3 years. That is a very long time to wait for the results of a single longevity experiment. On the other end of the spectrum, nematode worms live for weeks, which is convenient, but they lack the complex organs, adaptive immune systems, and vertebrate brains that define human physiology. The African turquoise killifish has emerged as the missing link. It combines the complex anatomy of a vertebrate with the short lifespan of an invertebrate. The utility of the killifish lies not just in the speed of its aging, but in the fidelity with which it mimics human biological decay. An aging worm does not develop heart disease or kidney failure in the way a human does. The killifish, however, shares our complex anatomy and, crucially, our genomic vulnerabilities. One of the most striking features of this fish is the instability of its DNA. Its genome is packed with what are called tandem repeats. These are repetitive sequences of DNA that are notoriously difficult for cells to maintain. In Nothobranchius furzeri, these repeats make up nearly 21% of the genome. This is a staggeringly high number compared to other vertebrates. This genomic clutter creates what scientists describe as a house of cards architecture. As the fish ages, its cellular machinery struggles to replicate these unstable regions. This leads to DNA damage and genomic instability, which is a primary driver of aging in all species, including humans. Furthermore, the killifish offers a unique window into telomere biology. Telomeres are the protective caps at the ends of chromosomes, often compared to the plastic tips of shoelaces. As cells divide, telomeres shorten, eventually leading to cell senescence or death. This is a key mechanism of human aging. However, mice have notoriously long telomeres that do not shorten in the same way ours do, which limits their use in studying telomere-driven aging. The killifish, remarkably, has telomeres of a similar length to humans, around 8 kilobases. As they age, their telomeres erode rapidly, leading to the same kind of cellular exhaustion seen in elderly humans. This makes the killifish the premier model for testing telomerase therapies and understanding diseases like dyskeratosis congenita. At the cellular level, we also see mitochondrial burnout. The mitochondria are the power plants of the cell. and their dysfunction is central to the aging process. In the killifish, mitochondrial function plummets as they age. They produce less energy and leak more toxic reactive oxygen species, or ROS. Recent studies have shown that the expression of genes involved in the mitochondrial respiratory chain drops significantly in older fish. This mirrors the metabolic decline seen in aging human muscles and brains, providing a perfect platform to test interventions that might boost mitochondrial health. However, the story of the African turquoise killifish is not just about decay. It is also about a miraculous ability to pause time. While the adult fish is a model of rapid aging, its embryo holds the secret to suspended animation. Because their ponds dry up, the eggs must survive in the baking mud for months or even years until the next rain. To survive this, they enter a state called diapause. Diapause is not merely sleep, it is a complete metabolic arrest. The embryo pauses its development, lowers its heart rate to almost zero, and stops growing. Crucially, it stops aging. An embryo can remain in diapause for a year, which is twice the lifespan of an adult fish, and yet, when the rains return and it wakes up, it grows into a normal adult with a normal lifespan. The time spent in diapause essentially does not count against its life expectancy. Research led by Stanford University has begun to unravel how the embryo preserves its cells so perfectly during this stasis. The answer lies in epigenetics, specifically a protein complex known as the polycomb complex. Scientists identified a specific gene, chromobox7 or CBX7, which is dramatically upregulated during diapause. CBX7 acts as a master repressor. It binds to DNA and shuts down genes related to metabolism and muscle development, effectively putting the cell's engine into neutral. It prevents the accumulation of cellular damage that typically comes with the passage of time. When researchers used CRISPR to knock out the CBX7 gene, the embryos could not maintain diapause. Their muscles withered, and they burned out, unable to survive the long wait. This discovery is profound because humans also possess the polycomb complex. The machinery for cellular preservation is encoded in our DNA, lying dormant. If we could learn to activate CBX7 or similar pathways in human organs, perhaps in a donor kidney waiting for transplant, or in neurons threatened by stroke, we might be able to induce a localized pause that prevents damage. The brain of the African turquoise killifish is perhaps its most valuable asset to medicine, specifically regarding neurodegenerative diseases. Unlike the mouse brain, which is resistant to many forms of human neurodegeneration, the killifish brain is surprisingly fragile in a human-like way. Consider Parkinson's disease. This condition is characterized by the accumulation of a protein called alpha-synuclein, which clumps together to form toxic Lewy bodies that kill dopamine-producing neurons. Creating a mouse model of Parkinson's usually requires heavy genetic engineering to force the mouse to overproduce the protein. The killifish, however, develops these aggregates naturally. As the fish ages, researchers have observed the spontaneous formation of alpha-synuclein inclusions in its neurons. Furthermore, the specific populations of neurons that die in human Parkinson's, the dopaminergic and noradrenergic neurons, also degenerate in the aging killifish. This offers a clean model to test drugs that might dissolve these aggregates or protect neurons, without the confounding variables of artificial genetic manipulation. Similarly, the killifish is shedding new light on Alzheimer's disease. Alzheimer's research often focuses on amyloid beta plaques. The killifish possesses an APP gene, the amyloid precursor protein, which is highly conserved. A groundbreaking study created a knockout killifish strain lacking the APA gene. The results were counterintuitive and fascinating. The mutant fish without the amyloid protein had reduced cell death and performed better in learning and memory tests in their old age compared to their normal counterparts. This suggests that the wild-type amyloid protein, even before it forms massive plaques, plays a role in the natural aging and cognitive decline of the fish. It provides a platform to study the earliest, subtlest stages of Alzheimer's pathology, the silent phase that occurs decades before diagnosis in humans. The speed of the killifish model also allows for rapid drug screening. A prime example occurred in 2026, when researchers turned their attention to SGLT2 inhibitors, a class of drugs originally designed for diabetes, such as dapagliflozin. Clinical trials had shown these drugs protected the heart and kidneys, but the mechanism was unclear. Using the killifish, researchers could treat the animals and watch their kidneys age in real time. They found that the drug did not just lower blood sugar, it fundamentally rewired the metabolism of the kidney cells. It mimicked a state of nutrient deprivation, tricking the cells into activating survival and repair pathways similar to autophagy, which are normally turned off by the abundance of food. The treated fish maintained youthful vascular density in their kidneys and avoided the fibrosis typical of aging. Because the experiment took only months, the data was available years faster than a comparable mouse study would have allowed. Looking forward, the African turquoise killifish is becoming a technological platform for high-throughput vertebrate aging research. With the killifish genome fully sequenced and CRISPR-Cas9 gene editing fully optimized for the species, we are entering a new era. We can now create hundreds of different mutant strains, each missing a different gene, to screen for longevity genes in a way that was previously only possible in worms or yeast. We can test thousands of drug compounds for anti-aging effects in a time frame that matches the pace of modern pharmaceutical development. Furthermore, the killifish bridges the gap in regenerative medicine. While zebrafish can regenerate their hearts and fins indefinitely, killifish, like humans, lose this ability as they age. A young killifish can repair a fin injury, but an old one cannot. By comparing the gene expression of a young healer versus an old non-healer, scientists are identifying the specific biological breaks that turn off regeneration in older tissue. In the end, this tiny fish offers a humbling lesson. Biological time is elastic. It can be compressed into the frantic 4 months of the killifish life, or paused indefinitely in the suspended animation of its embryo. By studying Nothobranchius furzeri, we are learning to master the dials that control this biological clock. This research may one day help us buy the one thing humans have always craved: more time. Thanks for listening to G Fun Facts Online.